My name is Emad Moeendarbary. I'm a professor of cell mechanics and mechanobiology at University College London, Department of Mechanical Engineering, and currently applying engineering and physics concepts to understand the biology of the cells, how cells behave in the context of development, physiology, and diseases. The human biology is very complicated. You have trillions of cells interact in your body, and it's so difficult to understand how these cells are interacting. They are in 3D dimension, they are surrounded by different cells, and so complicated to understand them. So biologists and let's say people who are studying human biology and diseases try to understand the behavior of the cells in the context of health and disease inside lab. And these assays, the simplest assays, are like culturing cells from your body inside some petri dishes, and then you can look how cells behave, but also you can see how drugs and different therapeutics gonna be basically impacting the cellular behavior. So these are called in vitro assays. This means like we have in vivo or human trials, and we have in vitro assays that are like studying biology inside the lab out of basically animals or human. This is the definition of in vitro assays. The scope of this specific project is around like myelin and myelination, which is one leg of any neurodegenerative disease. So any kind of neurodegenerative disease is associated with myelin disruption. Neurodegenerative diseases are the diseases that are related to loss of function and behavior of neurons in the central nervous system. Neurons are the cells that are basically telling you to move, to think, to act. And these cells are central for basically your function, the daily function, thinking, cognitive, sensation. And so in neurodegenerative diseases, the function of these neurons are basically getting disrupted. And this is what we call neurodegeneration, because neurons degenerated. And the context of my specific research is around myelin, that are these sheets that are wrapping around neurons and helping neurons to be stable, healthy, but also transmitting the electrical signal in a faster way and more efficient way that when neurons are isolated. So these are the, like, let's say some wraps, some protection around neurons. And this protection gonna help neurons to transmit electrical signal faster, but also more efficient, but also keep neurons happy and healthy. They're like supporting cells to neurons. So this research is around understanding, developing advanced in vitro assays to replicate this process inside the lab and understand what are the mechanisms, but also understand if when this process is failed, if we can find treatments to improve this process. We developed an assay inside our lab. This assay, we call it micropillar assays, or we can call it artificial axons or artificial neurons. So just to make a definition, neurons have these extensions that are called axons. And these are the extension of neurons. We call it axons. So from now on, when I'm talking about axons and neurons, I may switch. But basically, these axons are the ones that are myelinated. So in the lab, we made artificial axons or artificial neurons in the form of pillars, micropillars. And then we put the cells that are responsible to making those myelin sheaths that I just explained. These cells are called oligodendrocytes. And in the lab, we make these artificial neurons, and we put the oligodendrocytes, and we observe that these oligodendrocytes are myelinating the pillars. Now the question is like, are these artificial neurons replicating the physical and biological properties of neurons? And the advancement that we made is we are claiming, or we are like proving, that our artificial pillars are replicating some of the most important properties of neurons in terms of both mechanical properties but also biological properties. We created this assay inside the lab, and the aim is to make it more replicating neurons, but also to understand the processes that are involved during this myelination that I explained. And the second step, or the ultimate aim, or let's say towards the end of the project, we would like to test treatments, including drugs, including genetic and other kind of treatment, to understand, to test whether we can enhance this process of myelination around neuron when it fails in neurodegenerative diseases. This is extremely highly interdisciplinary research at the interface of neuroscience and engineering and nanotechnology. Just considering those neurons or axons that I was talking in the central nervous system, the length, the diameter or size of these neurons, the thickness of these neurons range from 1-2 micron to 15 micron. These are very thin. Just imagine your hair has a thickness of 100 micron. These neurons are 100 times thinner. So making such a thin structure that replicate biological and mechanical properties of neurons is extremely challenging. So we use microfabrication technologies at UCL, at London Center for Nanotechnology, for creating those kind of pillars with that dimension. The first point is dimension and physical properties. The second point is like we are using biology and biochemistry to give these artificial axons the properties that neurons have. So we use microfabrication technology, we use advanced mechanical characterization technique, we use biology, biochemistry, and ultimately also we put these oligodendrocytes and put them inside our assay to wrap around these axons. And this is also neuroscience. We are working across disciplines and techniques across disciplines. Neuroscientists, or let's say neurobiologists, studied this oligodendrocyte behavior that are cells that are wrapping around neurons in a dish, in a flat substrate. So the flat substrate or the dish doesn't represent the three-dimensional thin nature of these neurons at all. So fundamentally, you cannot test drugs or let's say understand the process of making myelin sheets in a let's say unrelated, or we call it non-physiological condition. So they are using these stiff petri dishes. They are stiff, they are very, very tough and flat. And so this oligodendrocyte just spread out. They don't have any structure to wrap around. So the advancement, or the let's say the leap forward for our technology is the use of these three-dimensional structures. And this is something that is like novel. The most important part is we can tune biomechanical properties of these artificial pillars or artificial axons in a way that we want. So we can tune the stiffness, mechanical properties. We can tune the size, the length, the diameter, and we can tune what to coat around these artificial axons. So we have full tunable system that potentially replicating the real axons of neurons in the best way possible. The potential long-term impact from fundamental research is we want to understand this process of wrapping. It's extremely difficult to look at this process in animals or in humans, to see how the cells wrapping around neurons. This is not possible. It's very, very complicated. So there is like tens of thousands of questions about this process fundamentally. Because we want to cure diseases, first we need to understand what are the fundamental aspects of the process. The second leap is, once we understand this, we would like to know if we can improve or inhibit the failure of this process that is happening in neurodegenerative diseases. So we are like, our ultimate goal is to see if we can come up with therapeutics and drugs that we can screen in our assay, and this is gonna be basically accelerating research for drug discovery and treatment.